Thank you, Dr. Pont. Appreciate y'all having me to come speak today. I think what I'm going to talk about may sort of fit in with some of the ideas that have already been presented this morning. But I wanted to come talk to you about a protein that I'm very interested in, something I've spent about the last 20 years working on, and that is sequestosome 1, or called P62 for short, and talk a little bit about how it functions at the mitochondrial level and then relate that to some of the behaviors that we associate with dementia and Alzheimer's disease. So just to start things off, I saw this slide on the internet and I kind of liked it because it presents a number of the different symptoms that we can associate with dementia. Memory loss, irritability, delusion, things like that. Are very symptomatic of what we see with dementia in patients, and the most common form of dementia in the world today is Alzheimer's disease. So just a few quick facts about Alzheimer's. Just speaking in the U.S., it is currently the 6th leading cause of death in the United States. And this is very significant because the 5 in front of it, the cancers, the HIVs, things like that, we're getting better at. We're able to treat those a little bit better, so the deaths occurring from those diseases are not happening at the level they once were. Alzheimer's disease, however, is just the opposite of that. It's going in the other way. We're having more and more deaths occur from Alzheimer's. Currently in the U.S., there are 5 million people living with Alzheimer's disease. That number triples worldwide. And when we project to the year 2050, in the U.S., that number reaches 15 million. In the world, that number approaches about 45 million people living with Alzheimer's disease. And 1 in 3 of those subjects, one in 3 of those patients will die from some symptom, some offshoot of Alzheimer's or dementia. Monetarily, it's even a scarier proposition. Last year in the U.S., alone, over $600 billion was spent in the treatment of dementia. Again, you project that forward to the year 2050, and that number in U.S. dollars is $4 trillion. That's just in the U.S. Dementia numbers worldwide are closer to 50 million now and are expected by 2050 to reach almost 150 million people. When you do the math on that, it's staggering the amount of money that's going to need to be spent to deal with dementia-related problems and diseases. So what can we do about that as scientists, as base-level scientists at this point? Well, the ideal thing to do would be to find some way to possibly predict this disease occurring. Can we see, is there something out there that we can look at today to determine whether or not you may have Alzheimer's, or you may be developing it now, or you may have a disposition to it in the future. And what I'm talking about is a biomarker, okay? And biomarker is just a fancy word for any characteristic that we can measure, that we can get some sort of measurement on, that is an indicator of a biological state, okay? In the past and even now, Alzheimer's disease, in particular, is diagnosed with histochemical plaques. This requires some sort of brain tissue biopsy to go in where we stain for it and look at the occurrence of the plaques here. I'm sure everybody has seen something that looks like that. We're developing more and more technologies where we can use imaging to look at live brains, that we can go in and see possible deposits developing there. But we also want to look at the biological ways that we can address this problem to possibly find that biomarker, that characteristic that we can look at. In the pathogenesis of neurodegeneration, then, to look for that biomarker, no matter what trigger we look at, whether it's aging or stress or whatever, basic cellular primary responses to neurodegeneration are all going to pretty much be the same, and they're going to be some sort of development of a protein aggregate, whether that is breakdown of the UPS system, the ubiquitin proteasome system in the cell, or just protein aggregation. The garbage systems in the cell are not going to work the way they should. And this leads to the pathologies that clinicians and scientists have seen from anything from the Lewy bodies of Parkinson's disease to the fibrillary tangles associated with Alzheimer's disease. And these specific pathologies then lead to very specific symptoms occurring at the base level of gene transcription all the way to changes in how organelles in our body function. Now, the interesting thing about this, all these different pathologies, again, are plaques. They're aggregated proteins. And all of those proteins contain the protein that I'm interested in, P62. So just a little bit about P62. It was originally defined as a scaffolding protein. It doesn't have any enzymatic function, but it does have all of these different protein binding domains that allow it to interact with numerous different proteins in many different signaling pathways. And all of these pathways are in some way associated with some sort of neurodegenerative pathology. Now, P62 is most recognized by a couple of domains at each end of it, either the PB1 domain or the UBA domain, having it involved in those garbage systems that I talked about, either ubiquitin proteasome or endomitophagy. So it is very tied in to many of these different pathways that lead to what we see with neurodegeneration. Now, I'm interested in the mitochondria. P62 has been found to play a role at the mitochondria, some work that I've done that I'll talk about today, and others. But more specifically, in like Parkinson's disease, once you have a mitochondria and that mitochondria becomes defective, it begins to increase levels of ROS, or reactive oxygen species, which cause that oxidative stress to occur in the cell, as well as then deregulation of protein phosphorylation. We have the ubiquitin proteasome breaking down. All of these different things can be related to defective mitochondria. And again, P62 is involved not only at the mitochondria, but also in all these other systems as well. So P62 shows the potential to be a biomarker for neurodegeneration, and specifically into Alzheimer's. So I wanted to look at how P62 functions at the mitochondrial level and relate that later on into some behavior modification. I started this with tissue culture cells using mouse embryonic fibroblasts. And these are just stained mitochondria using MitoTracker Red that you put in to see the morphology of what a mitochondria looks like. Well, in normal cells, you get what's called a tubulo-reticular pattern. These mitochondria are nice, long, ribbon-like structures. They're not what we see in textbooks all the time with a little kidney bean and stuff, but they're nice, long, tubular structures. If you have a complete genetic knockout of P62, these cells do not have the P62 gene in it, the structure totally changes. It becomes very punctate, very rounded. When you add P62 back to this knockout background, so this is re-expression of P62, mitochondrial morphology is recovered. We go back to that tubulo-reticular pattern. If we count those and look at the individual different morphologies of mitochondria in these cells, you can see that wild type and re-expression of P62 basically give the same morphology. So P62 plays some role in mitochondrial morphology. Well, the structure of the mitochondria leads to function, and we all know that the mitochondria is the energy source of the cell. It's where we make ATP. That's what feeds and drives all the systems in the cell. It's also the place where we produce ROS, the reactive oxygen species I talked about earlier. In small levels, that's a good thing because ROS is a signaling molecule as well. In large quantities, ROS can become very toxic to the cell. So if we measure the amount of ATP produced by mitochondria lacking P62, you can see that it's about half the amount of normal cells. When P62 is re-expressed, we recover ATP production. And when we look at ROS levels, as you might expect if you don't have P62 in there, ROS levels are up in knockout cells. P62 re-expressed in those cells bring those ROS levels back towards a wild-type phenotype. So not only does P62 regulate morphology, It also affects the mitochondrial metabolism and energy production. Another way you can go and look at mitochondrial health is to look at its genome, to actually look at the DNA that's produced inside the mitochondria. Again, we measure the amount of mitochondrial DNA present, and P62 knockout cells have about half that DNA. When we re-express P62, you can see that it goes back from here to here, about the same level of mitochondrial DNA being expressed. The transcription factor, the mitochondrial transcription factor, TFAM, is also very important to mitochondrial function, especially mitochondrial DNA. It's required for mitochondrial DNA replication, transcription, and also performs a number of repair mechanisms to it to keep the mitochondria healthy. When we measure the amount of TFAM present in mitochondria isolated from wild type and knockout cells, you can see that knockout cells have a good bit less TFAM present in their mitochondrial DNA. There's some import problem there. And when we re-express P62 in those same cells, we recover to about the same level of wild type. So P62 appears to have some role in protecting the mitochondrial integrity, the mitochondrial genome integrity, by some TFAM-related process. Now, what was really interesting about this set of experiments right here is when we did them, not only did we re-express P62 in the knockout system, we overexpressed P62 in the wild-type system. So we took wild-type cells that had a basal level of P62, and we overexpressed P62 in them. And when we did that, what we noticed is that both mitochondrial DNA levels as well as the amount of TFAM present in the mitochondria increased with more P62. So overexpression of P62 improved mitochondrial function as measured by mitochondrial DNA. And this was above and beyond what basal or wild-type levels of P62 generated. Now, knowing this, I wanted to then look and see, was there some way we could go about measuring to see what effect this overexpression of P62 had on behavior patterns and relate that back to mitochondria. And this led to the development of a P62 overexpressing mouse. Now, I had done some work with some collaborators of ours previously, and they had developed a P62 complete genetic knockout. This mouse, as you can see, and this really is not the best representation of it, this mouse is extremely obese. It's much larger than a wild-type mouse. But again, total knockout of P62 leads not only to Alzheimer's or dementia-type problems behavior-wise, but also leads to some metabolic disease problems as well. But as I said, by looking at this P62 knockout mouse and looking at mitochondria isolated from hippocampal neurons from this cell, from this mouse, we can see that, as you might expect, the knockout mouse has the same morphology that our genetic knockout cells had, with very punctate and rounded. When we looked at mitochondrial function, looked at the ATP produced as well as the ROS, you can see that knockout mice, the neurons from their hippocampus, perform the same way that we saw with the tissue culture. So our P62 knockout mice did exhibit significant levels of mitochondrial dysfunction. When we looked at behavior patterns, because we've now got a model we can look at and see how this mouse performed, and we looked at a couple of different levels of dementia-related diseases, both anxiety and depression, you can see that the knockout mice exhibited a significant increase in the amount of anxiety and depression compared to the wild-type mice. So not only do we have the mitochondrial dysfunction, but we also have changes in behavior patterns similar to what we see with Alzheimer's disease. So I decided to make this transgenic mouse that overexpresses P62, but I wanted to get away from the metabolic side of things. I didn't want to have the metabolic problems that the P62 knockout mouse has. So the mouse that I generated only expresses P62 in the brain. Okay, so I used a Thy1 expression vector, inserted a human P62 gene so we could differentiate it from the mouse. The gene is tagged with an EGFP tag so that we can see it. And when we look at the expression of that transcript in this overexpressing mouse, you can see that in different brain regions, that P62 is certainly overexpressed, specifically in the hippocampus and brainstem region, learning and memory region in the hippocampus, but no other tissues express P62. And because of the EGFP tag, we can take hippocampal slices and actually see P62 expressed under the microscope. So we effectively expressed our EGFP tag human P62 in the mouse brain. And then we started asking questions about the mitochondrial metabolism that we see here. So again, harvesting hippocampal neurons from these overexpressing mice, the morphology of the mitochondria stayed about the same. We still had that long tubulo-reticular pattern that we have seen previously. And when we measured mitochondrial functionality, we had an increase in the amount of ATP being produced in the mitochondria from our overexpressing mice, as well as an increase in TFAM levels in the overexpressing mitochondria. So P62 overexpression gave us all the hallmarks that we might have expected from improved mitochondria functionality. More ATP, the genome is better because we have more TFAM present in there. So our overexpressing mouse model appeared to work well when we were just looking at mitochondria. So then the question became, does this improvement in mitochondria affect, or mitochondrial function, affect any positive changes in brain behavior associated with neurodegeneration? Now, what kind of behaviors are we talking about? We've already had some discussion on that. We're talking about things like anxiety and depression. Clinically, this is called affective spectrum disorders, things like panic attack, irrational fears, things like that. So these are the types of behaviors that I wanted to look at. The first behavior I looked at was the anxiety of these overexpressing mice, and I did it using an open field maze. The open field maze can be used for a number of different behavioral testing paradigms, but I was looking at anxiety here. So this was the maze. We could test 4 mice at a time. Basically, you put the mouse in the middle of the maze for a period of time using a suspended camera up here and computer tracking software. You watch the mouse move, okay? And the first thing we found was that the total locomotor ability of the mice was the same. So the overexpressing mice and the normal mice moved about the same pattern in the maze. So there really was no change due to overexpression in the motor ability of the mice. When we looked at the express tracks of how the mice moved in the maze is where we started to see our differences. And the wild-type mice tended to spend more time around the outer wall of this open field maze. This is stigmataxia. Feels safer being up against the wall as opposed to being out in the middle of a big room where something can find it and possibly eat it. So the wild-type mice spent more time in the outer part of the maze as opposed to the inner part, whereas the overexpressing mice were about 50-50. They didn't mind getting out here in the middle and moving around and exploring their environment a little bit. So while their locomotor ability was the same, the amount of anxiety that the overexpressing mice exhibited was less than what we saw with wild type. I also tested depression in these mice using a version of the pour salt swim test. And this is basically a test where you take a mouse, you put it in a beaker of water, he can't touch the bottom, he can't climb out. And you measure his activity for about 6 minutes, looking at what he does during that time, and you're looking for when the mouse gives up. When does the mouse say, okay, I've had enough, I'm done? This is the immobility time. Now, When we measured this between the overexpressing mouse and the wild-type mouse, there was no difference in when the mouse sort of gave up the ghost, or whatever term you want to use here. When they just quit swimming was about the same. However, when we looked at the individual behaviors being recorded during the test, and we measured swimming and climbing ability, we saw that the overexpressing mouse spent significantly more time just swimming in the maze than did the wild-type mouse, while the wild-type mouse spent significantly more time trying to escape the maze. The mouse was panicking. The wild-type mouse was panicking at this point. He wanted to get out. He was scared. Whereas the overexpressing mouse was just sort of out for a swim. Okay? So we interpreted this as that the amount of depression that the overexpressing mice, depression and despair that the overexpressing mouse showed compared to the wild type was significantly less. So overall, in affective spectrum behaviors, overexpression of P62 seemed to improve these different behaviors in our mouse model. I also wanted to ask some learning and memory questions. How well did this mouse learn? Again, these are symptoms associated with dementia and Alzheimer's disease. First, I did long-term potentiation, and again, as Dr. Pant referred to, this is basically taking a hippocampal slice from the brain, stimulating in one point and recording in another point, and seeing the time it takes for that stimulation to reach from point A to point B. In the knockout mouse, we can see that it performs significantly less than did wild type. So if you don't have P62 in your brain, you don't learn as well. Your ability to learn decreases. When P62 is overexpressed in the brain, we see a very significant increase in the ability of this mouse to learn. The LTP time is much better. So overall, we saw improvement in LTP. I also did this testing using a non-invasive method, looking at hippocampal function called a Barnes maze. This is a circular platform. It has all these holes in it. One hole has an escape box. The mouse is put in the very center of this maze. The room is very brightly lit. There's a very loud noise going off in the maze or going off in the room. The mouse wants to escape that. And we're asking over a period of days how long it takes for the mouse to learn where his escape box is, where he can get in there and make all the light and the noise go away. Over the acquisition phase, over the training phase, learning phase, wild type and overexpressors appear to perform about the same level. You might say the overexpressors maybe learned it a little faster than the wild type mice did, but maybe not a whole lot more. However, when we went to asking questions about how well they remembered, we did probe trials. And probe trials are simply repeating this test for one instance, one test, removing the box and asking how long that mouse spends in the correct quadrant or correct area of this maze looking for where the box should be. Our first probe trial was done 2 hours after this time point right here. Okay, so this is their short-term memory. How well do they remember it? Very short term. And wild type and overexpressors performed about the same. They both remembered short-term memory where their escape box was. However, when we did probe trial number 2, which is 7 days past this, the mouse has not been exposed to the maze for 7 days, okay? And we performed our probe trial on day 12, we can see that the overexpressing mice performed significantly better. They remembered over a week's time where that escape box was. So while we didn't see any real changes in short-term memory, we did see a very large improvement in long-term memory with P62 overexpression. So overall, our learning and memory overexpressing P62 improved, as well as our affective spectrum disorder behaviors. So in conclusion, P62 affected not only the mitochondrial functionality, how well mitochondria perform, but also behavior patterns associated with different neurodegenerative diseases. And we believe this places P62 as a biomarker that we could look at for prediction of possible neurodegenerative diseases, and then somewhere down the line, someone coming in and doing some targeting for potential drug therapy, drug discovery, and everything related to P62 and P62 overexpression. This is our mouse. It is currently available, currently housed at the Jackson Laboratory, and available to the scientific community now. And these are my collaborators, lab mates. I thank y'all very much for the opportunity to come speak.
Thank you. Time or questions?
Sure.
Wonderful talk. Go ahead.
Yes, ma'am.
Was the overexpressor, did it reduce the body weight back toward control?
It stayed about the same amount. The body weights of the overexpressing mice are about the same as wild-type mice. Yes.
I'm curious as about AMPA-K.
Mm-hmm.
It's like glucose metabolism, and did you measure the changes in glucose metabolism to see if
We did not. That has not been done yet with the overexpressing mouse. Our collaborators have done that with the knockout mouse, as obviously effects there. But no, it has not been done. We haven't done that with the overexpressor. Yes, sir.
How long does a mice live, and what happens to the normal mice when they grow older?
In our colony, mouse age would reach, this is a black 6 background mouse, they would reach 18 to 20, 24 months on the outside, but about 18 months. As far as what happens to them when they age, I haven't done the aging study to go back and measure everything yet. Phenotypically, both wild type and overexpressors will increase in body weight just a little bit, but as far as their behavior patterns, they stay about the same. Overexpressors. So
All these diseases are diseases of the old age, isn't it? And most of these neurodegenerative diseases happen when you grow older.
mhm.
So don't you think that it is part of the aging process which affects more than anything else?
You mean the overexpressing the
No,
No.
the dementia process and all that. There's a variability in the normal aging process in human beings, which is causing this problem in the first place.
Right, but what I believe is that overexpressing, yes, aging is very much a factor in all neurodegeneration, all neurodegenerative diseases. As we get older, our bodies don't work the way they used to. We're not, all those garbage mechanisms and things like that aren't kicking in and doing what they should. But I believe that looking at P62, that we can show that it improves that mitochondrial function and could possibly then come in and improve it in an aged individual as well.
Yeah. He's always comparing with the wild type. So what happens, sir? Is there any more questions? What happens? What happens the knockout under similar parameters?
Under similar parameters, the knockout always perform worse than the wild type.
Do you see any plaques or tangles?
Yes,
You do.
yes. They have very much a similar AD type phenotype, similar to the 3X transgenic mouse.
So if you cross them, what will happen?
We're working on it. Exactly. As soon as someone wants to kick in, we will be glad to jump all over that.